thank you all for being here in our seminar series. I'm delighted to welcome, I can say now, my former colleague, Bernardo Lemos from Harvard University, the School of Public Health. We've been working together for quite some time on various projects, so it's not going to do any service to Bernardo if I spend a lot of time introducing him. You have his bio sketch and CV online anyway. But just shortly, Bernardo graduated from Harvard evolutionary and population genomics, and then joined the School of Public Health and founded environmental health at Harvard School of Public Health. He's leading the lab there on environmental epigenetics. He's doing a lot of things in terms of environmental stressors, well known in the field. You know, I'm intrigued with his title of presentation. You know, it includes everything, not only environmental exposures and stressors, and very recently his research is focusing on aging, which is another emerging area, especially as it linked to the environmental exposure. So with that, I will welcome him again, and thank you all for making the time, meeting with him, interacting with him, and Bernardo, the podium is yours.
Thank you very much, Phil. Thanks for the very kind introduction. Thanks, Nancy, for the invitation to be here today. And yes, I'll be talking to you today, but I'm really representing those guys, the people in the lab who are really essential and did all the work. It's really an honor to be talking about what they do. And what we care about in the lab is environmental epigenetics in the context of aging. And what that is the study of the impacts, the consequences, the mechanisms by which environmental exposure, be it pollution like you see there, be it lead painting, peeling from a wall, there's a child nearby, be it chromium to which those workers, welders are being exposed to, be it microplastics, their impacts, their consequences, the mechanisms by which they influence the epigenome. And whenever we can in the lab, we try to take an epigenome-wide view to try and gather data whenever it is affordable and we're sufficiently engaged to ask questions across the whole genome. But while we're doing that, we focus or we anchor a lot of our studies in the biology of this one organelle that sits inside the cell nucleus that is called the nucleolus. Can you all hear me well? Great. So let me just introduce you to the nucleolus a little bit. So what you see here in this picture, that black blob at the center there of the cell nucleus, that's called the nucleolus. And what the nucleolus is first is an organelle, right? So it's an organelle inside the cell nucleus. And it's also the site of biogenesis of the ribosome. It is the place from which ribosomal RNAs are expressed. So let's just talk a little bit more about this picture. So you have like DNA around the nucleolus, so all the chromosomes will be placed in the cell nucleus, somewhere in there, in that light shaded, I lost my pointer. So you can see the light shaded area around the nucleolus. That's where the chromosomes will be. Now there is also DNA that goes inside the nucleolus. That is the ribosomal DNA. Okay, this ribosomal DNA is responsible for giving origin to that organelle, and it's also the place from where the ribosomal RNAs are expressed. So you think about a gene. Any gene that you're interested in, most likely is going to be in that sort of light gray area around the nucleolus. It's going to be sitting there, it's going to be transcribed, the mRNA goes to the cytoplasm. In the cytoplasm, that mRNA meets a ribosome, and that ribosome executes the process of translating that mRNA into proteins. Now that ribosome comes from where? It comes from the nucleolus. And what happens then to the protein components of the ribosome, the ribosomal proteins? They're also being transcribed elsewhere in the nucleus, not in the nucleolus at high levels. They're also translated by the ribosome. You have then a ribosomal protein that comes back into the cell nucleus, goes inside the nucleolus, and there it is assembled.
So what did you think?
That is assembled.
Can you mute, please?
It is assembled together with this nascent ribosomal RNAs.
Mm-hmm.
So one thing about the ribosomal RNAs, they're very highly expressed. So if you think of any gene in the genome, it is very lowly expressed relative to the ribosomal RNAs. The ribosomal RNAs represent over 70% of all the RNAs in a cell. So there's a whole lot of energy, a whole lot of transcription that happens from inside that organelle. Now, that's the background on the ribosomal RNAs. They're essential, they're very conserved, they're essential components of the ribosome. Now, what we've known for decades really from structural studies, that nucleolus is also disrupted or altered or modified in a variety of conditions. So if you take cancers, for instance, there's a whole literature on the nucleolus being modified in a cancer. It can be maybe in some reports a bit large or broken down, or the morphology looks a little unusual. There's also been reports of nucleolar alterations in neurodegeneration and in cancer. And that's sort of where it's an organelle that I really love. And there's a whole biology around it. It's being mechanistically linked to aging, the process of aging itself, to stress response, to metabolism. You can imagine the nuclear architecture, so how the chromosomes place themselves inside the cell nucleus depends also on the placement of that black ball in there. So the nuclear architecture is influenced by the nucleolus. So what we're going to do today is to first talk about biomarkers of aging. I'll define what they are. It's broad-based biomarkers of aging, and we're going to talk about mechanistic biomarkers of aging. And we're going to also look at the ribosomal DNA clock that we developed in the lab, and we're going to look at brain aging and Alzheimer's, and we're going to look at environmental exposure. So we're going to cover quite a bit. Most of the talk will be on aging and ribosomal DNA clock. I'll give you some case study of research we're doing in Alzheimer's, and we're going to finish with notes on environmental exposures. Now, so the part one, biomarkers of aging, and let's talk about those first. So why age? Why do we care about the study of aging? And really, if there's anything you'd like to know about someone to ascertain that person's risk of disease, and there was no time that this became more clear than now, I guess, in the middle of a SARS-CoV-2 and COVID-19 pandemic, is age. Age is the single most important risk factor predicting disease and mortality. We know that. We also know 2 and 3. That's, most of us know that, but I just want to repeat because they're just so fundamental, and you just forget those things all the time, right? We don't pay attention to them. Aging is manifested widely across the tree of life. There is extreme evolutionary conservation. We age, rats, mice age, fruit flies age, C. elegans age, single-celled yeast ages as well. Moreover, the single intervention that is most impactful to slow down aging, calorie restriction, also works across the tree of life. So you give primates fewer calories, maybe don't know how happy they are, but there are studies that show that they do look better as they age. If you give mice and rats fewer calories, they live longer. True for Drosophila, for C. elegans, true for yeast. In some studies, 30–40% higher longevity just by virtue of decreasing calorie intake. So what we'd like to do is to decouple, because we know intuitively, right, that there is a decoupling between chronological and biological age. There is, we know by I think intuitively really that the people age at different rates. And what we'd like to know is to decouple that and have ways to measure that decoupling. So we see there on top is a person of a certain age, and you see there in the red line the chronological age. That person on top has an accelerated biological age clock, and it's going to present with age-related comorbidities early in life at higher number. The person at the bottom, on the other hand, will be someone that is aging at a slower pace. It will present with age-related comorbidities later in life and smaller numbers. So I'd like to be able to decouple that and understand that process better of aging and biological age in particular. And for that, what we need are biomarkers, right? I think we do need really important biomarkers, and I would like to just take a moment here with us and discuss what are the requirements for a very relevant, a more relevant biomarker of aging. And I would call that the first requirement is that the biomarker should be mechanistic, and I think that's true for a lot of what we do, right? So if you think about a biomarker, we'd like it to be more than just wrinkles on the skin or the graying of a hair. We'd like it to speak to a certain biology, a certain mechanism, an underlying process in the cell that relates to the mechanism of aging. We'd like a biomarker to be predictive. Predictive of what? Predictive of chronological time. We'd like a biomarker of aging to change and progress over time, because we know time is passing. If there is a biomarker of aging that doesn't change with time, that is awkward. We'd like it to be conserved across a variety of species. for the reasons we're seeing, because aging itself is conserved. We would like it to be responsive. To be responsive to what? To interventions that impact longevity. So we'd like these four properties to be present in a biomarker of aging. So what we're going to do is first ask the question, do we need improved measures of age and aging? And the answer is yes. Traditional markers are not really great, accurate age timekeepers. They are often not that well linked to health and disease or the mechanism of age. And depending on the biomarker we're talking about, of course. And here are some examples. There is DNA damage biomarkers, there are markers of cell senescence, but we're going to focus on telomere features and telomere length. And why? Because that is the most widely used biomarker, and it's really widely used in human population studies. It's also used in the lab, in cell studies as a way of ascertaining aging. And let's try and think about telomeres from the perspective of these four criteria. And why telomere is so widely used? Because it is really mechanistic. The biology of telomeres is fantastic. For as a matter of definition, telomeres are those caps at the end of chromosomes. There are no way you can have a linear chromosome without a telomere. Otherwise, you have to have a circular chromosome. The mechanism is fantastic. There's been a Nobel Prize given to the biology of the telomere, the discovery of the telomerase, the enzyme that makes telomeres become longer. Telomeres are extremely conserved, which is also their value, right? So telomeres are conserved. The concept, even the telomere themselves are not. So some organisms got rid of telomerase. They don't have telomerase anymore, but they still make telomeres in ways that they had to reinvent. So it's very conserved, very mechanistic. We know what it does to a cell. If you lose telomeres, the chromosomes become glued, they link to one another. There are all sorts of problems that happen, and over time they tend to erode, diminish. So we know their biology, and that's, I would claim, that's why they got so much traction. The problem is they're not really that predictive telomere length. And if you look, I'll just give you one example here of one study, and I'm not trying to pick on them. There are a variety of studies like that, and what you're seeing there on the x-axis is age, chronological age. And what you're seeing on the y-axis is relative telomere length. And one thing to note is that, yes, telomere decreases with age, and that's been shown again, and again. However, there's a whole lot of variability. And there is no guarantee, if you were to measure telomere length in this room, that after 20 years there will be a decrease in telomere length, because we're in the middle of the pandemic, this is not very interesting talk, we don't, there is a certain number of people, we need a large population to get that result to be true. If we were to measure our telomeres today, and 10 years from now, I can guarantee some of us will have a longer telomere. So there's a lot of variability, and that is a bit awkward. That said, the biology is fantastic, and it's really, of course, a worthwhile endpoint in any case. Now, what I'd like to discuss with you are those epigenetic, DNA methylation-based, what they call broad-based biomarkers of aging. So, and I would claim here is that they're not mechanistic, and they are not conserved, but they are very, very predictive, and that's in essence why they over time became so popular in my view. Before we go into more details on them, let's just take a look at the history here. So it was about 2013. Two human DNA methylation clocks were invented by two different groups, or a group and a person, and they were invented at the same time. Really published almost the same time. The one that's lesser used was published a bit before. What they identified was that by measuring DNA methylation across the whole genome, and DNA methylation are, just as a matter of definition here, we're talking about methyl groups, CH3 groups, chemical groups. They're placed on the backbone of DNA, and they play a role in genome architecture. They play a role in gene expression. So you can measure those DNA methylation marks across the whole genome, and one group identified 353 CpGs. They were sufficient, just by measuring them on a person's blood, sufficient to predict the chronological age of that person. And a different group found that, if you use a different set of 71 CpGs, you would also be able to ascertain someone's age. And that's how they came initially. But there was nothing special biologically about these CpGs that made up this clock, and they were not conserved between the studies either. So that was a bit awkward. Those CpGs that's why I call them broad-based. They were coming from all sorts of places across the genome. Some are in between genes, some are within genes, some are in the middle of nowhere. there was no clear rhyme and reason for why you should use them on a clock. Now, it was about 2017, 2018 that, I mean, first this research grew a lot in humans, the biological age clock in humans, and 2017, 2018, a few groups started measuring DNA methylation clock in human cohorts, in mouse cohorts, excuse me. And the first study, what they found was that there were 99 CpGs that were sufficient to predict chronological aging in the mouse. Now the problem was those 99 CpGs were different than the ones they found in humans, but they said they have the same property. They were coming from all over the genome, and there was no clear rhyme and reason why they should go together into a clock. Yet they were able to predict the mouse age. So that's a little bit of the history, and it's been really a big challenge to find the mechanistic basis for those clocks, and clearly the clock itself is conserved, but the CpG that make up them is not. A little bit more of the history. Those are the two on top. Harvard clock and Hannum clock were the two first invented. The mouse clock came next, and in the last few years, just before the pandemic, there were two also clocks developed that became over time more popular. And one thing that's common about these clocks is that they're developed mostly using blood or multi-tissue, several tissues across the body, and they were broad-based. All right, so this is, but now let's just take a moment and see how well they work, because it is, to me at least, quite impressive that you have a correlation like this. And what you're seeing is on the x-axis chronological age, and what you see on the y-axis you say DNA methylation age. And you have correlation coefficients in both cases here, 0.85. This one is studied, but again, this is. I'm not picking this study. There are other studies also with that tight a correlation between chronological and DNA methylation age. In this case, this study used two different clocks, and you can see how tight the correlation is. So it's actually quite, it's quite amazing that one can, by taking a piece of blood, measuring DNA methylation marks. I mean, those chemical groups, CH3s, on top of cytosine groups in the DNA backbone. By measuring that, you can come up with someone's an age correlation that's that good between chronological and DNA methylation age. So that's a bit of the story, and that's when, and I would like to argue with you that we came in and developed this clock. We didn't know it was going to work, that we called the RDNA age clock. That I would argue has a mechanistic component to it. It's predictive enough, it is conserved, and it is responsive, in this case, to exposure as well, or to interventions that delay age or accelerate age. So let's talk a little bit about this. So what's the work here that we've done? We're going to assess RDNA methylation age in a mouse aging cohort. We're going to assess RDNA methylation in blood, in a cohort of 255 mice from 16 age groups, about 15 animals per age. The old animals were 36 months old, and all animals in this cohort are maintained in a diet that they can eat as much as they want. And we're going to separate those animals after measuring RDNA methylation. We're going to separate these animals into a training set and a test set. And we're going to train the clock in the training set and see if the clock performs reasonably okay in a test set. And we don't know. We're going to ask the question, can an RDNA clock be trained? Does RDNA age approximate chronological age? And that's what we're going to do first. And what you see here in this slide, and that was very exciting when we first seen this, is that there is a very tight correlation between chronological age on the x-axis and RDNA age on the y-axis. What you're seeing there in blue, those are the training animals in which the model was trained. And you see two metrics of performance on top. One is the correlation coefficient between chronological and biological age, and the other one is MAE, mean absolute error, is the sort of the deviation between the chronological and the RDNA age. And it's reasonably low, 2 months, to about 2 to 3 months of error in a cohort that goes up to 36 months. So there's a very nice correlation in the training set, but also true in the testing set. Animals that had never seen the clock were able to ascertain their RDNA age and match that RDNA age with chronological age. So that was very, 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 very exciting to us, and obvious that it was going to work. Now, what we're going to do is to ask the question, can we also show that this RDNA methylation clock is responsive? and responsive to what? To an intervention that we know should slow down age, and that is calorie restriction. So we're going to take 16 mice from 4 age groups, 4 animals per age, and maintain these animals in the calorie restriction and ask the question, is there any signal that the rDNA age is slowing down? And that's what you see in here. So what you see here is the same data you've seen before, first in black. In black is the correlation between chronological and rDNA age. In black you see all the animals, and you see them also not, you see the individual points. And it's notable that they all fall on that nice line, and there's a tight correlation. But there in blue is the calorie-restricted animals, and you see the slowdown of their rDNA clock. So we're very, very, very excited with this kind of behavior for the biomarker when we first observed. Now the question is, okay, this is working, seems to be working fine in mice, but is that can we make this work for other animals as well? And one thing to keep in mind is that, again, this RDNA segment of the genome is extremely evolutionarily conserved, like very, very conserved indeed. I mean, it's 100% conservation between humans and mice for most of the segments in the RDNA. So we're going to then apply this, this clock to rats. And we're going to take rats that were fed a controlled diet or 2 dietary interventions for 6 months. And we're going to assess biological age when they were 11 months of old. And here you have the controlled diet, dietary intervention A and B. And ask the question, is there an impact of the dietary intervention on age? And what we've seen is that the control diet, we're able to, after data normalization, predict the right RDNA age. And what you see notably there, diet A and diet B, both diets reduced the RDNA clock significantly. And if you look at, this is in the aggregate, but if you look at the individual animals, you also see that, well, first thing is that not all animals age the same. Even in the control diet, there was some variability, right? Here you have some animals that were supposed to be 11 months of age. And they were the ones that were the most young. The one that was most young out of those 12 had an age of 7.727. Now, when you look at the dietary interventions, though, you see some really huge drops. We were very, very surprised. So there are animals there that show a 57% reduction in biological age by virtue of being given this dietary intervention. So we're excited that the dietary intervention, so the impact of diet was being reproducible in another organism, but also that their clock like this was operational in a rat.
Are the rats housed individually or as a group?
They were housed not completely in a mix of both. Yeah, they're not in single cages, but they're not all together. Yeah, yeah, but there was some nesting there. So the question now is, can the RDNA clock operate in humans? And that's the kind of picture we see. The answer is yes. There's a very nice correlation between chronological and RDNA age. And now the question is, can we also apply this kind of clock to cells? And what you see here is cells from mice, and you see fibroblasts from kidney and lung, and you see induced pluripotent stem cells. And you can what you notice that by doing producing the pluripotent stem cell, there is a notable decrease in the RDNA age of these cells. So this, we still have quite a lot of work to do, in my view, on cells, but that is a good indication that this clock operates on a cell level as well, in a dish. So the question here is now, okay, there is a clock, an RDNA clock, and it works reasonably well. So the question is, why does it work? And I'll say the reason, I think, one of the reasons is because there's an abundance of age-associated CPGs in the RDNA. And let's just take a look here for a second. What you see here, just think about sort of the making of these clocks. These clocks are composed of CPGs, and CPGs that you take from the RDNA or you take somewhere else in the genome. So what you're seeing here is a distribution of correlations between a certain site, methylation level, and age. You see in blue. So for sites that are coming across the genome, you have, in this case, we've done this for both humans and mice. For this case, a picture of over 4 million CPGs in the mouse genome. And what you see first is that most CpGs don't care. They're around 0 there. So what that means is that as these animals age, the methylation doesn't go up or doesn't go down, with any regularity. There is no correlation between change in DNA methylation and the status of that particular cytosine in the genome. Okay, methylation level is either stable or changing erratically. So there's no correlation. Now, when you're looking at 4 billion CpGs, there's a fair number of CpGs that fall on the ends of this distribution. Many of which will have positive or negative correlations with age. So the animal is getting older, and that methylation level in that particular CpG, for whatever reason, is going up or going down. And when you're sampling across the whole genome, you're just going to pick those ends of the distribution. You're going to make sort of many different versions of a clock, if you want. Now, we weren't clear that we were going to be able to produce a clock for the rDNA because we were then looking here at a much smaller segment of DNA, much fewer CpGs. But the reason partially why this clock was able to be produced was because there was an enrichment for CpGs that are tightly linked to age. So if you look at the rDNA in particular, there's a number of CpGs in there that are associated with age, and that was very nice. Now we're going to just take some moment here because there's so much data in this distribution. I just want to take a moment to ask the question: what might be happening at the extremes of this distribution? Is there any signal for interesting biology there? And one thing we noted is that there is an excess of mitochondrial genes with negative age-associated DNA methylation. Okay, so here's the question. Here you have genes that are transcribed inside the nucleus. They localize them to the mitochondria and to the mitochondrial inner membrane, and there's 23 times more CpGs in those genes that are negatively correlated with age than positive correlated with age, and a highly significant result. So what that means is that as these animals age, and us as well, this result was true both in mouse and humans, as they age, there was a loss of DNA methylation in mitochondrial genes. So that's, just to give some nugget of what else is happening in this sort of kind of system. And if you look in the other direction, there seems to be an increase in neuronal genes. So there's as these both mice and humans age, there's a tendency for neuronal genes to gain DNA methylation. They're becoming more and more enriched in DNA methylation marks. For genes that belong to a class of learning and memory, there's 6 times more positive associations with age than negative associations with age. What that means again is that these neuronal genes, as we age, normally, and humans and mice, they tend to accumulate a higher density of DNA methylation, and that has implication because what is happening normally that these DNA methylations are shutting down these genes. So that's just normally what the data seems to be showing. Okay, so now we've seen some of these clocks. What I'll try to do now is to give you some flavor for the application of these clocks to in the context of a human population study, to see how, is there any meaning in these clocks really, and can they be useful? And here in particular, in the context of Alzheimer's. So we're going to look at brain aging and Alzheimer's in this cohort called the ROS-MAP cohort. Okay, that's the religious order study of memory aging project. These are 2 longitudinal clinical studies that were started in 1994 and 1997. There were over 1,400 participants in them, about 300 of them diagnosed with Alzheimer's disease thus far. There was no Alzheimer's disease or related dementia at entry in this study, a diagnosis. And people were entering this study at about middle age, okay? And now, this is a really terrific cohort. We're lucky to be working with them. And what they do, these subjects, when they enter the study, they agree to also donate their brain for further analysis. So when they pass, their brain is examined for signs of pathology related to Alzheimer's disease, but also others, and so nicely documented. So what we're going to do here is to look at DNA methylation and layer measurements in 689 post-mortem dorsolateral prefrontal cortex, DLPFC, samples. And we're going to take those DNA methylation marks across these brain samples and calculate those, what we call broad-based DNA methylation ages. And we're going to use 4 clocks that, 2 of the original ones, 2 that were developed more recently, but also one that was developed with cortex samples. It's called a cortical age clock, very, very recently, developed using another set of brains, but specifically in brain samples, and also apply them in the context of this cohort. And we're going to ask questions like, okay, is there a relationship between accelerated aging or accelerated biological age as measured by those clocks and Alzheimer's disease? Is there an association between Alzheimer's disease pathology? And that's an interesting aspect too, which I wasn't completely clear when I started this study. There was some debate there, so we'll see a little bit here. And the results are not identical. So here's first there's an association between broad-based biological age clock acceleration and clinical Alzheimer's diagnosis. Okay, that relationship is not there for 4 clocks, but for the 1 clock that was developing brain, there was. For each standard deviation increase in cortical age clock, there was a 54% greater likelihood of clinical Alzheimer's disease diagnosis. And we're talking about clinical diagnosis here. Now, what we're going to do is to apply and test whether there is also an association with pathology of Alzheimer's, which is interesting because it's a fascinating field. The clinical diagnosis doesn't go hand in hand necessarily with the pathological diagnosis. So, and these are done by different individuals. There are the clinicians. There are these people. The subjects participate in this study. They every year come back to the clinic and get an array of tests that will tell the clinician whether or not they have a signs of Alzheimer's. That's fantastic. But once they pass, those brains are also examined, and then there's someone else, the pathologist, who will tell, without knowing whether that brain was a pathological Alzheimer's or not. And there is not, the agreement is far from perfect. And what we see here is that for the Alzheimer's pathology, there was a nicer association actually for across the board. For 3 of the other clocks, they were again developed in blood and multi-tissue. There were associations. They were very, very, very. The results were very similar. For each standard deviation increase in biological age clock, there was a 30% greater likelihood of a pathological Alzheimer's diagnosis. But then for the cortex clock, which is kind of intuitive too, I mean, for the clock that was developed with brain samples, this association was much stronger. So for each standard deviation increase in clock age, there was a 90% greater likelihood of pathological ID, Alzheimer's disease. Now, then what we did was to break down not just for not only using the metric, pathological Alzheimer's disease or not, but look at the specific, the components of the pathology, amyloid beta and tau tangle density. I won't go into any details on those, but just to say that they were also individually associated with accelerated age. So a higher biological age in the brain of these patients was associated with a higher pathology as well at the individual pathology level. So now, of course, we're very interested in understanding what's going on in this context from the perspective of the RDNA and the nucleolus, and that's a lot of what we engaged in right now. And I want to now take a turn and finish with notes on environmental exposures. I think will be speak especially to us. And I was trained as a geneticist over time, complete transition into environmental health. I think there was so much. It's so underappreciated, the role of environmental exposures on diseases, and especially I still have many colleagues in genetics and even the epigenetics field has to learn a lot from exactly the environment and the impact of those or whatever the environment is on biological systems. So I'll just present some notes, some data on chromium, on lead, and on microplastics. Okay. So what you see here is a very simple experiment. Is exposure, chromium exposure. We've done chromium exposure in lymphoplastoid cells derived from blood lineages, as well as from, in this case, BIS2B bronchoepithelial cells. And what we're doing here, the protocol is straightforward. We're doing a 24-hour exposure to chromium in these cells. At 24 hours, we remove chromium and continue passaging those cells, growing them in a petri dish over actually much longer now than 7 days. And we've done this across a range of concentrations. And we're going to ask the question, for the purposes here, what's the impact of chromium exposure on genome-wide gene expression? Are the genes responding? Is DNA methylation even responding? I might have removed the DNA methylation data. Yeah, so the answer is yes. There's the cells care if there's chromium in the median, and we've done this across a variety of concentrations. But what's interesting here is that there's both up response, like genes are being upregulated, there are genes downregulated, but the response is sustained even though the exposure itself only happened for a 24-hour period. Okay, you see there at day 7, and what you're seeing, first you have the hours, 0 hours, 24 hours, 3 days, 7 days, and you have log fold change relative to baseline. So we've done a control at the baseline. And you see here the red line going up means that the gene is going up. A red line goes down, it means the gene is being downregulated. The genes that are in red are significantly up or significantly down. The genes that are in gray are not changed significantly. So yeah, you see both up and down, but you see that it goes up and stays up, or goes down and stays down, even though the insult only really happened in the interval between 0 and 24 hours. And you see this from the methylation data as well. I'm sorry I removed the slide, but I'll just briefly say here, if you measure because these are mRNA levels, so these are genes going up and down, but there's an underlying epigenome that is responding as well, that presumably is helping maintain those genes up and maintain these genes down even after the insult was removed. And then from the DNA methylation perspective, it's actually quite nice. You have almost no change from 0 to 24 hours. Even a little bit between 24 hours and 3 days, there's very little change. But then 3 days, then there's all this sort of reorganization. You see a lot of change in DNA methylation that happens between 3 and 7 days, if I recall. We can look later for those slides. And it sustains for 14 days. So 14 days after the insult, we still see change in the DNA methylation landscape that were caused from these events 14 days prior. So we're very excited this kind of pattern that we see more and more in our data and others. Now, another exposure I wanted to present to you as well is lead. We've been over a few years now collecting preliminary data on that. And here in the context of lead exposure and neuronal differentiation. So the experiment is straightforward. It's kind of like the other one as well. You're taking neuronal progenitor cells, differentiating them, and we're doing this with Collaborator at UCSD, differentiating them into full-fledged neurons, and in that process, giving them very early on some, a small dose of lead, and then we have profiling and trying to understand what's happening with the genome and the epigenome. And what we see is that, like I said, whenever we're doing this, we're taking a broad perspective, we're recording all these events across the whole genome, but I always try and zero in on the genes that I care the most for, and those are the genes of the ribosome biogenesis pathway, genes that are related to the nucleolus, and we do see a variety of candidates, in that case RPL-L23, RPL-L29, 19, S5. These are all components of the ribosome. These are structural components of the ribosome that are changing up or down as a function of because of the prior exposure to lead. And in this context, we're also able to measure some changes of sort of larger changes in these organoids, these brain organoids that were related to the lead exposure. So we're very, very excited with this kind of data. Now, I wanted to finish with some data here because I know many of you are interested in microplastics. That's something that we've been doing in the lab over the last 5-6 years now. This is an interesting project. We were not, I was telling Phil we're not able to fund it, and we didn't even try. We were trying to just fund it from within the lab based on other things. I think we all do that. We're also good at that at some level. But it's been an interesting, exciting field to just try and support and give it some, maybe some preliminary data to it. In this particular case, we are looking at microplastics, and it's of a certain kind. There's a polyethylene microplastic. They're virgin. So, and we can discuss, there are many caveats to that. They are of a certain size. In this case, it's about 50 micrometers, and it's a certain dose. In that case, 10 to the 4 particles a day given to these animals. And we're going to ask the question, okay, giving these animals microplastics, are there changes in the genome and the epigenome? And we're going to, in this particular slide, focus on the gut and ask the question, not from the epigenome perspective, but the mRNA levels. Are they changing up and down? And we see a few things. One is that, yes, there appears to be an impact of microplastics, at least at that dose, that size. We see genes responding up and down by virtue of those animals being prior, their prior exposure to microplastic, and the exposure happened at that dose for about a month. Now, what we also saw that was very interesting, we did joint exposure with DEHP, with a plasticizer, and I won't go into the details of what it does, but you can find this in the water. Most of us have some exposure to DEHP. But what you find that was curious was that if you gave DHP alone, there was a certain effect. If you give microplastics alone, there was a certain effect. But if you give them together, the effect was so much more enhanced. It's so much more enhanced. So you can see things like here, in oxidative response. These are genes that differentially regulated. You can barely see anything happening with DHP, barely see anything happening with microplastics. If you give them together, there was this really enhanced impact. And we see this again and again in our data, and not just for DHP, for other toxicants as well. So I'll give you an example, and this is with cadmium. And in that case was mice. This is with fruit flies. So what we have here is exposure of fruit flies to cadmium and to microplastics. And in this case, of a certain dose, a certain size, the size was 5 micrometer, the doses, we did a dose response, a variety of doses. It's hard to think of those of microplastics in the context of flies, so I'm not even going to go there for now. We can have that conversation another time. But we're going to look at what's, is there anything happening with the data? And what you have here first is this gut damage assay. So what you have here is larvae or fruit fly. They are given a controlled diet without microplastics or microplastics plus cadmium. And what you see there, that blue dye, is a marker of gut leakage and gut insults. In this, so it's a really, really nice system. It works really well. We've done, of course, all sorts of controls. We've given microplastics alone, we've given cadmium alone, and we see some gut damage in those systems as well. Especially, even cadmium alone does damage the guts, depending on the dose. But if you put them together, it is higher. And then we run these flies through a climbing ability assay. So it's an assay of locomotor function. If these are size, and we've done this in the lab. At that time, before the pandemic, we had fruit flies in the lab. And it was a very, very motivated graduate student, Marina Volovska, and a postdoc, Jan Zhang, and they really did a lot of work on this for this one. And they were able to show that there was a relationship between the ability of these flies in this climbing assay that we had to develop. Others, we took from the literature, but we had never. And I was excited to see the data work because they really showed. They really did a good job, and went out of their way to try and make it work. And that's like an assay of putting flies to climb, essentially, and in a particular context, and we measure their ability to do so. And you see with control flies, they just go up. That's what they do. So if you throw them to the ground, they're going to go up, and they actually walk up in a vial. And there is a variety of different treatments, but I want to call your attention here. So if you do microplastics and cadmium, they really lost their ability. They look like dizzy flies. It was very strange. And those are flies that had experienced gut damage early in life. Now, finally, there's this assay here. This is an assay that speaks to me in particular because, doing epigenetics, being interested in gene regulation, gene silencing. What you see here is a normal fruit fly eye. It's red. And what happens to the red eye and it's a particular model. Just think of the red eye as a marker of the expression of a certain gene. And this is a gene that is just poised in a certain way that it can be, it can be shut down or not. And if it's not shut down, you have a nice red eye. That's the gene operating fine. But if it gets shut down, whatever mechanism there is, the eye becomes white in this particular way. And you have here the control fly, you have the microplastics, you have already some signs of gene silencing, gene inactivation. If you do the cadmium, it's actually quite strong just by itself, but the cadmium this effect, they both come together to a stronger effect in which there is gene silencing in the microplastic cadmium joint exposure.
Does that involve the zest white locus at all?
This involves the white locus. Yeah, it's like a white mottled 4. Yeah, it's an inversion that's like a, yeah, I can get it. This is exciting. So since you asked, I'll take your time. So it's an inversion that places the gene close to a segment of heterochromatin, a segment that shuts down the genome. And since inversion, if the gene is next to this, so imagine that all this genome here is shut down. That's what it is. That particular part of the genome is off. And you place the gene very next to it. So any sort of perturbation that makes that genome, the area of silencing expand, will shut down the gene. And when it's shut, and this happens developmentally, and when it shuts down the gene, you have this sort of variegated phenotype, because there is a quantitative aspect to it, but also qualitative. So these eyes are they're ocelli, so they're all independent. You can have like a certain one shut down completely, another one shut down less so, and it creates this. Yeah, that's exactly right. Okay, so I'm going to close. What we've seen today are biomarkers of aging. Just notes and remind us what we've seen. Biomarkers of aging. We talked about broad-based and mechanistic biomarkers of aging. We talked about the ribosomal DNA clock. I hope I made you a little bit excited about that. We love those clocks. I've shown some research we're doing on aging Alzheimer's, environmental exposures, and I think there are, at this point, really huge opportunities for environmental epigenetics as a field as a whole, and for in particular also epigenetic RNA aging clocks, in mechanistic studies, in animal studies, in translational research. There's a lot that needs to be done, and in particular in the context of environmental exposure. I want to go back and give credit also to Amanda. This study, she was like an amazing. She loved her draws and produced this beautiful picture, which shows you the cell nucleus, all the chromosomes around it. You see the nucleolus there in the center. It really is something that we carry deeply in the lab and always go back to one way or another. And I want to finish with thanks to collaborators and people, all the people who are in the lab essential to this work. I highlighted there in blue the people whose data I showed today. We've seen data from all of them at one point or another. They've been terrific. Thank our collaborators and thank our funding sources that have been fantastic over the years as well. So with that, I want to finish here, and we have time for questions. Thank you.
Thank you very much for this great presentation. I don't know how we're going to do this, but why don't we open the floor for questions from the audience here, and then we will go on Zoom, and then they can ask their questions. So, questions from the audience.
So in the experiments where you're doing the caloric restriction, since methylation is your endpoint, are you sure that the animals are not just deficient in methyl donors?
Yeah, that's an excellent question. That's an excellent question. No, we're not sure. That is a possibility. That is a possibility. That is a possibility. So the question is, if I may say more, the methyl group, the certain methyl donor that's SAM that's given equals to provides to the methylation machinery to put these marks in there. So we're not sure that could be the case. But we tend to think of this experiment, even though it wasn't measured, the methyl group availability. We don't tend to think of it as a lack of methyl donors, but you're right. That should have been and that's a good question because it's typically not measured in calorie. So calorie restriction now as a field became an exercise on trying to modify the protocol. Also understand the biology, but trying to modify the protocol. Why? Because the protocol of calorie restriction is at some level kind of unfortunate. If you tell people not to eat, it's not as nice. So people are trying like intermittent diet, all sorts of things. So that's one path. And the other is like trying to understand the mechanism. Understand the mechanism became much more of a challenge. But it's thought of as something beneficial, and because of methyl donor availability, when you don't have enough of it, that's thought of as something bad. Say that's why you have Broadbased in pregnancy. You want to have. There is the assumption there's plenty is better. So that's a good question. It's not looked at enough, and we certainly did not in this context.
Okay. Right.
So you've got a great measurement of aging with ribosomal DNA, and you showed that with calorie restriction you can decrease the chronological or the biomarker of aging versus chronological. And then you went on to environmental exposures. My question, does environmental exposures increase chronological or rDNA age?
Yeah, and we have some evidence. So very good question. So just back up a bit. So in humans, this been being done by many other groups besides us, and being done in humans in particular because those clocks, interesting enough, they were invented in humans first, and then they came to other organisms because people were kind of annoyed by not understanding mechanisms. They were invented in humans. So in the human literature, there are plenty of links between There are some really solid papers out there that show, exposure to air pollution accelerates biological age, for instance. Yes, so the link is there. Now, in the animal world, there is no such a data, not nearly as close. So there's a lot of work that we can do there. That needs to be done. In cell systems, there isn't. We have some of our cell data suggested, yes, that there is an impact. We've seen some in cadmium, cadmium treatment, and in case of the collaboration of animals, cadmium may be decreasing biological age. Again, a certain dose, again, a certain period, you have to. When you're doing this experimentally, there is a lot more variables that you need to play around and dig deep into. So the answer is there. In animals, we don't know so well. In cells, I have good indication that that's happening. Again, conditional on protocols, and there are all these tweaks that one needs to worry about. In humans, there is associations that are truly believed to be true. Yeah, and that's for the broad-based.
And that's with the ribosomal DNA.
So the human ones are for the broad-based clocks.
Right.
We haven't about
ribosomal.
Yeah,
DNA.
we haven't tested ribosomal DNA. Yeah, thank you.
But also, just on the same note, I like what you show there, that it's complex mixtures. And I think when we start using the real world complex environmental mixtures, then you will start seeing the effects. The synergistic effects. And that's, or at least the in vitro studies and all these simplistic models, they miss that part. And it's more literature right now showing that the synergistic effects are there. Okay.
Yeah, I'm curious about cells that are multinucleolated. So how do the multiple nucleoli coordinate with one another, or do they, in these cell responses?
I don't think we know the answer to that. So back up here too. So we're thinking about cells often with one nucleolus. You're talking about cells that are sort of like syncytial multiple nucleus or multiple nuclei?
Well, I'm thinking, let's just take a hepatocyte, which may have 6 nucleoli, or a large neuron, a Purkinje cell, may have 4. And are they responding in coordination, or are they responding in competition, or
we don't know
is their response so heterogeneous that...
No, we don't know the answer. No, what we've seen, and that's interesting, and that goes back to the other question too, we don't know the answer to that. We've seen in some systems, in Chromium for instance, there is an increase in nucleolide number. If you treat them with chromium, what I see is that there is a breakdown. I mean, again, there is some variability there, but say if there is a cell with 1 or 2 nucleoli, and so the result is highly significant. If you give them chromium, again, a certain protocol, you see I don't know if the word is breakdown, but you see an increase in the number of nucleoli. Now, the question is how they're communicating with one another. I can't speak to that. We haven't done that.
Curious, when you look at the aging process, do you see a change in methyltransferase expression levels? Do you have like higher methyltransferase methylation?
Yeah.
Curious.
I don't think we do. I specifically look at some of them. Methyltransferase 2, no, I don't think we have that. No, I don't. I mean, I was trying to think also from the perspective of the DNA methylation marks, if around the DNA methyltransferase. The mRNA, I don't think changes with age, no. I think that's the answer that I have for you now, from what I remember of the data. Which is, so that's an interesting question. So the methyltransferase, there is a whole machinery there placing those, taking those methyl groups and putting on the DNA methylation, DNA backbone. I don't see change with them with age.
Dorothy.
So the data you showed in the mouse aging study, you were using blood or plasma, I believe. Did you look at any of the tissues to see if the slope of aging of our DNA changes in high turnover tissues like the intestines versus the brain or something?
Yeah, I would love to see that data. We don't have it. Yeah, we don't have that. Yeah, we are. Yeah, that was in blood.
So if there are no other questions from the audience, we'll open the floor for the...
That's an excellent question, though. I'd love to know.
the online. Yeah. Any questions from the online people? Please go ahead, identify yourself and ask the question. I think they're muted.
Unmute yourself. Unless Maria's
I
controlling.
think Maria probably muted everybody.
Oh, because.
No, I didn't. Everybody's able to unmute themselves.
Okay, thank you, Maria.
Great.
All right, I mean, we just don't know.
I don't have a question myself, but I see that the chat has several questions. If anyone
So I
wants
could
to
read
check the
it.
chat.
Maria, can you read the question?
Can you read? I don't have access to the chat room, so...
Oh, okay, sure. Let's see. I have one. First question is there evidence that some clocks work better at the beginning of the lifespan while others are more accurate later in life?
So there is one clock. So there are many aspects to this question. Really great question. There is one clock that's been proposed to work better at young ages. I didn't talk about this, but it's a broad-based clock as well. I use sort of CPGs all over the place. There is a claim for that clock to work better at younger ages. It's a very new clock. I'll just stop there. I don't have enough to tell you. Now, what I can tell you also, that all the clocks, what they do is they don't do as well at old ages, right? So, and you can see that, and by not doing well, that's also kind of have to put some grain of salt there. So what I mean is that late in life, they tend to be, there's more error. But maybe that's fine, because maybe that's exactly what's happening. We're kind of younger, all in the same way or similar ways. As we age, there are different ways of aging, maybe, and it's expected to be a greater dispersion later in life. So that's true for a lot of clocks. So a lot of clocks, all the clocks that I've seen have this greater dispersion later in life. There's one clock that has been proposed to work best in young age, and that's it. But whoever asked that question, if you have follow-ups, or that's an interesting question.
I have
I'd love to
another
hear.
question
Yeah.
from the chat. Is organelle aging directly related to overall aging?
Organelle aging. I don't know. I don't know how do you measure organelle aging. Yeah, maybe whoever asked that question can reach out over email, please. I'd love to hear more about your question, because I think it's a good question. I'm less clear on how to assess organelle aging. Yeah.
Are there any sex differences between the various clocks?
Yeah, we did not see. Good question too. We did not see sex differences. No.
Okay. Any more questions, Maria, on the chat room?
No, on the chat, no.
That's cool. All right. Well, then we should...
One more question.
Sure.
So what's noted about the pattern of methylation in the ribosomal, and when it changes, does it change across, or are there different sections that change?
It changes. What we've seen so far, it changes across the whole thing. There was a segment of it that has a. So it changes across all of it. There's a segment. There are parts of it that have like just a baseline much higher levels than the others. So imagine like everything is changing, but then some have a baseline that's like here, the other one's like there.
Okay.
For some reason, some sites start at a very high level. They tend to go up to. That's
Okay, thank you, Bernardo, again for wonderful presentation. Thank you all for attending. Thank you all online. I'll see you in the next seminar, which is, I think, next week.